about the two concepts. One is immune and another is immunization. Immune is a person who is cured from the disease. Suppose a person get infected with hepatitis B infection, and in the course of the time he get overcome the virus, clear the infection from the body. Then in that person, if you go and watch out the antibodies level, there will be the antibodies of every portion of the viruses. So the person has immunoglobulin, like antibody against the surface antigen, antibody against the core antigen, antibody against the E antigen. So they have antibody against every antigen. And why that is? Because the patient has got infected with the whole virus, and now patient get cured, patient own immunity has overcome that infection, and you will found all antibody in that patient. But in case of the immunization, when you talk about the hepatitis B vaccination, a portion we give in vaccine only a portion of the virus, that is also not a live product. So a killed product of the surface of virus actually given to the patient. And what will happen? The antibody that will developed by the patient will be only towards that particular antigen, that surface antigen only. So when we give a vaccine, what is vaccine actually? Vaccine is nothing. Vaccine is one of the way to produce the antibody inside your body. That is why if you remember this COVID pandemic, where we have given lot of types of vaccine to the different type of people. Like we have given a Pfizer from Moderna, then we have this SARS-CoV-2 like from the Chinese vaccine, like and then there is the COVID shield from the UK, then there is this Covaxin, there are other like Varicella and etc. So the main purpose of this vaccination was to give you the virus, the component of the virus, so that you develop antibody against it, that component. And when actually the live virus attack you, then your body already have the virus antibody level. Now when live virus actually attack us, that antibody level that was produced due to the vaccine will protect us and not allow the virus to enter inside our cell, or say neutralize the virus, and doesn't allow the virus to make us sick. So the main vaccination is actually one type of giving virus to you. But the thing is that in vaccine, we doesn't give you the whole live virus or whole live pathogenic virus, high virulent virus. We give you the portion of the virus, or say by killing it or by altering it. So there are the different mechanisms by which we can make the virus low virulent, or we remove the toxicity. That can be given, even live vaccine is given, but that is attenuated one. We kill the virus, so the antigenicity is only preserved, but that is given to your body, so there will be the production of your antibody. And there are other vaccine as well. Okay, so hope you have understood the vaccine means giving you the virus or component of virus, but that component in the vaccine is expected or is given in such a way that you will not develop the disease, but your body will be sensitized, your body will be form antibody against that component. So when there will be actual live virus attack you, at that time your body has high level threshold, above the threshold level of antibody, and that will prevent that live virus to affect you, to enter inside your cell, to make you sick. So that is important. Okay, let's move to see what are the points that we need to understand. The vaccination is a true milestone of the medicine and has saved countless lives from the preventable disease. That is called immunization preventable disease, IPD. So that there are certain diseases that can be prevented from the immunization, like diphtheria, then there is the tuberculosis. There are a lot many diseases that can be vaccine preventable disease or immunization preventable disease. The concept dates back into the 11th century when the Chinese practiced in the art of the variolation. However, the practice is credited by the Edward Jenner in 1798, where he used a strain of cowpox virus to protect a child from the smallpox. So that was the initial start of the vaccination. This chapter will discuss about the science behind the vaccination as well as a summary of the type of vaccine currently used in medicine. So secondary and subsequent response. To understand this secondary and subsequent response, we'll talk about it, okay? But let's see what are the vaccination that can be given to the patient. So before understanding the vaccination, you need to understand the types of the immunity. Actually, we have the immunity that is divided into two parts. One is the active part, another is a passive part that can be natural and artificial. The main thing is that active means your own body is involved in processing that antigen and form the antibody. Whereas in passive, the body is not taking part in formation of antibody, so that is passive. Term passive is given, and that is actually given to you from the preformed form. The antibody is preformed and that is delivered from the outside. It can be naturally from mother to baby, or it can be from artificial, like rabies immunoglobulin, diphtheria immunoglobulin, or anti-rabies immunoglobulin. So these are the anti-diphtheria serum, anti-rabies serum, these are the anti-horse serum. These are the actually the component that is preformed outside. I would say monoclonal antibodies. These are the preformed antibodies that can be available in the market, and you can deliver to the patient when required. So these are already formed. There is no body active involvement in formation of that component, so that is called passive. Naturally, it can be acquired from the mother, from the placenta, that is immunoglobulin G comes, and from the breast milk, immunoglobulin A comes. And then coming to the that is passive but artificial, that will be this anti-diphtheric serum, anti-rabies serum. These all are your component that can be given. That is a preformed form that can be given to the person. Okay, talking about the types of immunity, we have talked about this passive, that is natural, placental transport, IgG and colostrum, and also we are talking about this passive artificial, that is the horse antivenom against the black widow, spider bite, snake bite, horse antitoxin against the botulism and diphtheria, pooled and human immunoglobulin versus hepatitis A and B, measles, rabies, varicella zoster, or humanized monoclonal antibody against the respiratory syncytial virus. These are the preform that is already available in the market, and you can give directly to the patient. So these are the passive. One thing is important to understand over here about the passive but natural immunity is actually we get from our mother, and the immunity of the mother is actually important to protect the baby in the initial period of life. In the initial period of life, when baby get delivered, he or she doesn't have the immunity for fighting the diseases, but there are the virus or bacteria in the environment, they have to encounter them. So the immunity that is transferred from the mother to the baby is actually the protective, only protective immunity to that inborn child. So a mother who is actually living, who is having, say, not living in a good condition, not living in a good condition means those who are exposed to many viruses, many bacteria, they will develop the disease, they will overcome the disease, and that antibody is already in her body, and that will actually transfer from the mother to the through the placenta to the baby. So the mothers, if you remember, in the slum area and in the poor community people, we see the women have exposed to a lot of this unseen microbes or pathogen. They have fighted and they have win and they have developed antibody inside her. So the baby which has been delivered from them actually have this antibody that has been transferred through the placenta or through the breast milk to the baby. And you can see the baby which is born in the poor slum dog or say poor environment, we see many are actually having no diseases in the initial period of life. Why? Because the maternal antibody is present inside that baby, and the mother has already fighted many diseases, and because of that, that antibody is still in that baby, and that is protecting that baby from other environmental pathogen. So the mother who has exposed to the different diseases is actually good for the baby. If she has win that diseases, it doesn't mean that mother should get exposed and should be died due to the disease, not that in that sense. She should be vaccinated, because vaccination will produce the antibody and that will be transferred through the placenta. She should have been exposed. The more she exposed to the disease and win the disease, the more antibody will form, and that will be transferred to the baby, and the baby will get the immunity when there was in the early infancy of the life. That is important. That concept you have to understand. Then talking about the natural, there can be active means the recovery from the infection. There is a clinical infection, clinical and subclinical infection, from where you can actually recover and you develop this, where your own body will take parts in the formation of antibody, and that is the natural active means. And there can be the artificial active means as well, that is the vaccination. When you give vaccination, like any vaccine, any component, hepatitis B, DPT, Haemophilus influenzae, polio, measles, mumps and rubella, varicella zoster, anything that is given to you, that the body will take as the antigen, they will develop the antibody, and after formation of antibody, the body has taken part in the role in formation of antibody. So initially the antigen will be there, that will be captured and processed by this antigen presenting cell, then that will go to the T cell will activate it, then it will activate the B cell, convert it into the plasma cell, antibody will form, and antibody will, there will the, if it is a protein component, then T cell involved, since there will the memory cell, and memory cell will be always there. So if there is a second, there is actually exposure to the viruses or bacteria, that will be cause the secondary immune response, and that will kill that virus or bacteria or neutralize that. So that is the main concept of your artificial active means, that is one of the type of immunity. So hope you have understood about the active and passive immunity. We have talked about this passive immunotherapy as well, where we are giving the preformed antibody. The problem is that introduction of antibody from the other species can generate IgG antibody, which may cause systemic anaphylaxis. If you have heard about the treatment of like diphtheria, we give anti-diphtheric serum, and anti-diphtheric serum is actually can if you don't give that serum to the baby or diphtheria patient, then patient is going to die due to diphtheria. On the other hand, if you are giving this say anti-diphtheric serum, this has the potential to cause systemic anaphylaxis and cause the death of the patient. So treatment itself can be one of the responsible for the death of the patient. So you have to be careful, because this anti-diphtheric serum, or say any antibodies, the preformed antibody, can even given to the baby that or a person that will generate this IgG antibody, which may cause systemic anaphylaxis. The generation of IgG after infusion within even human gamma globulin is particularly an issue in the person with selective IgA deficiency. As IgA is a molecule that have not encountered before, this patient can however given IgA depleted globulin. So we can remove IgA from that globulin and can be given to the person who have IgA deficiency disorder. Okay, so there are introduction of antibodies from other species can generate IgG or IgM anti-isotype antibodies, which form complement activating immune complex, leading to the possible type 3 hypersensitivity reaction. Introduction of antibodies from human can elicit response against minor immunoglobulin polymorphism or allotype. So that if the antibody is given from another person, so there will be antigen antibody reaction, and that will cause this complement activation and maybe lead to the type 3 hypersensitivity reaction. So that is also the issue of your passive immunotherapy. Now talking about the types of vaccine, that can be live vaccine, okay, that can be live attenuated vaccine, which means that the virus is actually live, okay, or the bacteria is actually live, but we have attenuated that virus or bacteria in such a way that vaccine, the viruses or bacteria in the vaccine will not cause the disease to us. They will only come in our body, they will replicate. Once they will replicate, they will attack the antigen. Our body immune system will kick on. They will try to clear that infection. The immune system doesn't know it is pathogenic or not. It's only know it is a foreign, and they have to kill it. It is the immune system has nothing role to decide it is pathogenic or not. They don't know that formula, or they don't have been trained in a such a way, so they know it is dead or alive. Even they don't know dead or alive. Even they don't know pathogenic or non-pathogenic. Anything that is foreign, they will develop the antibody. So live attenuated vaccine is attenuation of the live virus or live bacteria, but they do not cause the diseases. The toxigenic component of that bacteria or virus is removed. So this is called live attenuated vaccine. The compromise of live organism which lost the capacity to cause the disease, but still replicate in the host. So there will be large number and more antibody will form. West has stimulated both humoral and cell-mediated immune response. They mimic the natural infection and typically elicit the lifelong immunity. So the main aim is they mimic the natural infection. It seems that your body has been infected with the viruses or bacteria, but the thing is that viruses or bacteria doesn't have the toxigenic component. They cannot make you sick. They cannot damage your cell. They only multiply in our body, and our immune system make antibody against it, and then that will be protecting us from the future actually toxigenic virus or bacteria. Typically, 1 dose provide immunity, but 2 doses are used to ensure the seroconversion in most of the individual. Dangerous for immunocompromised patient because even attenuated viruses can cause significant diseases. Since attenuated vaccines are composed of live organism, there is still potential to revert back to the virulent form. One thing is very, very important to understand that any live viruses cannot be given to the immunocompromised host. This live viruses is contraindicated for the people who are immunocompromised, because although we have removed the toxigenicity, but still that low virulent can also be a potential pathogen for immunocompromised people, or maybe that will revert back to the virulent form and will cause the disease in severely immunocompromised patient. So for severely immunocompromised patient or immunocompromised patient, live attenuated vaccine is contraindicated. Okay, so live viruses viral vaccine we can recommend it in the United States, and actually you can remember with the formula, that is the Mr. Square VJ Psyche PSY. So this will be the MMRR, this R. Okay, so we are talking about MMRR. So this is MMR, this is rotavirus, then VJ varicella zoster virus, then A for adeno, and PSY this includes. So MMR, you have understood measles, mumps and rubella. So this is measles, mumps and rubella. Measles, mumps and rubella is live virus. Then is the rotavirus, that is also live virus. Then varicella zoster virus, that is also live virus. Adenovirus, that is also live virus. Then is the polio, Salk and Savin. So Savin for this will be the polio, then smallpox and yellow fever. So this polio will be actually, let's say, VIN, Savin component. So polio, Savin component is a live virus. Smallpox is a live virus. Yellow fever is a live virus. So these are the virus that is live attenuated virus. Adenovirus is also live attenuated virus. Varicella zoster is also a live attenuated virus. Measles, mumps, rubella is there. Rotavirus is there. So you have measles, mumps, rubella. You have rotavirus. You have varicella zoster. You have adenovirus. You have polio. You have smallpox. You have yellow fever. So this is my formula to remember this organism. You can remember in your formula, which makes you easy for remembering this organism. But you have to understand this is the Mr. VJ Psyche. That is a way to remember this that is live attenuated vaccine. So measles, mumps, rubella, rotavirus, varicella zoster, adenovirus, that is the polio. Which one is the Savin? There is another form, Salk, that is falls under the killed one. Then there is the smallpox and yellow fever. So these are the viruses that actually live vaccine that you need to remember for USMLE or any other medical exam or for your practical life uses in infectious disease as well. Now coming to the another concept of non-attenuated. That non-attenuated is sometime what we do, we can give, although it is a very rare scenario, we give actually the pathogenic virus to the person to develop the antibody. So in such a way, so if a person is healthy, we exposed that person with the pathogenic virus, not attenuated one. We have preserved the